The advantage is,
I think she's not
the
here.
advantage of that.
Yeah, thanks so much.
Is the portability. You can kind of optimize the parameter set on one instrument and just port it to another
I think we are good to go. So hello everyone. Today I have the pleasure to introduce Michael Skinnider. Dr. Skinnider received his undergraduate degree from McMaster University and an MD PhD from the University of British Columbia. And during this time, he was also visiting as a PhD student at the École Polytechnique Fédérale de Lausanne. After completing his PhD, he started his laboratory in the Levi-Sigler Institute for Integrative Genomics and the Ludwig Institute for Cancer Research at Princeton University. His research focuses on the application of machine learning to the problems in biology, chemistry, and medicine. His work has been recognized by a number of awards, including the Forbes 30 Under 30 list, the International Barnstable Award, the Dan Davis Scholarship, and the Borealis AI Fellowship. So without further ado, please help me in welcoming Dr. Skinnider.
All right, thank you very much for that introduction, and thank you for having me here today. I'm excited to tell you a little bit about what my lab has been working on very recently in terms of accelerating the discovery of new human metabolites using chemical language models. So sort of to give a high-level overview of what I'm hoping to articulate today, I guess there are sort of three key points that I want to communicate. The first is that using mass spectrometry-based metabolomics, it's currently routine to detect thousands of small molecules in any given biological sample, but very few of these are currently being identified. The second point is that, in my opinion, identifying these small molecules in high throughput, even with existing mass spectrometry data, is a very tractable and a very important computational challenge. And the third is that chemical language models allow us to reason about the scalar mass to charge ratios that we observe by mass spectrometry in a very new and very rich way. And I'm going to explain to you exactly what I mean by that statement. But I thought I would start first just by talking at a high level about why I think mass spectrometry-based metabolomics is so interesting, particularly as a computational biologist. And the reason is that I think it's remarkable that today we can routinely measure essentially all of the DNA, RNA, and protein in any given biological sample, thanks to the maturation of genomics, transcriptomics, and proteomics. And yet it's virtually impossible to measure all of the small molecules in that exact same biological sample. So when you put these side by side, it really becomes apparent that this is quite a profound gap in our ability to measure and ultimately understand the natural world. And it's been argued that this gap implies that the vast majority of the small molecules that you and I will be exposed to over the course of our lives are still unknown. And I think this gap is particularly striking because it's not for lack of an appropriate analytical technique. With mass spectrometry, we already have a technology that routinely detects, that is to say, collects data from thousands of small molecules in any given biological sample. And this data that mass spectrometry collects is very rich. It's very multifaceted. We're able to measure a lot of different properties of these small molecules. And so the challenge really is a computational one, I think. How can we take this rich data that's being generated by the mass spectrometer and link that data to the chemical structures of the small molecules that were detected by mass spectrometry? And this is a problem with a very long computational history going all the way back to the 1960s. And unfortunately, it turns out to be very difficult. And it's sufficiently difficult that today, in a typical mass spectrometry-based metabolomics experiment, well under 10% of the small molecules that are detected by mass spec will actually be identified. So what about this other 90% of unidentified small molecules? Well, this has been referred to somewhat evocatively as the chemical dark matter of metabolomics. And so putting this all together, I think that there's really an enormous opportunity to address this gap and illuminate this metabolic dark matter by developing new computational tools that can extract more chemical information from these routinely collected mass spectrometry datasets. And so today I'm going to tell you why I'm particularly excited about the advent of chemical AI and chemical language models in particular to help us tackle this challenge. So before I jump right into the meat of chemical language models, I wanted to, I guess, also talk briefly about how I came to be interested in this field. And this is actually an interest that goes all the way back to my very first experiences as a scientist at McMaster University, working with Nathan McGarvey. And we were working on automating the discovery of new bacterial metabolites using genomic and metabolomic data. And I think it's useful to sort of talk briefly about this because it really encapsulates the clinical or therapeutic value that computational mass spectrometry of small molecules can help realize. So the context here is that the vast majority of the drugs that we use in the clinic today are derived in some way from small molecules that appear in nature, many of which are produced by bacteria. And this is quite remarkable when you think of it. And in fact, since the turn of the millennium, with the advent of bacterial genome sequencing, these sequenced bacterial genomes have revealed that we've actually really only scratched the surface of bacterial metabolism. And so this might lead you to wonder, are there many other potential drug candidates encoded within these bacterial genomes? Now, unfortunately, actually leveraging these bacterial genome sequences to discover new metabolites or natural products, as they're often called in the setting of bacterial secondary metabolism, is still a major open question in the field. And so my work with Nathan was really focused on how can we combine genomic and metabolomic data sets in order to discover new natural products of interest. And the approach that we developed to overcome this gap works in two steps. In the first step, bacterial genome sequences are used to predict the chemical structures of genetically encoded metabolites. And in the second step, mass spectrometry-based metabolomics data from the same bacterium is then searched for those predicted chemical structures. So I'm going to talk through what each of these steps entails. This work goes back quite a few years now, and because it predates many of the advances in chemical AI that I want to talk about today, this entire platform is really predicated on the idea of codifying everything that we know about the biosynthesis of these natural products into a platform of rule-based algorithms. So what that means, for instance, is that this structure prediction module of this platform first searches the genome for all enzymes that are involved in the biosynthesis of these metabolites, and it encodes the exact chemical reaction that each of these biosynthetic enzymes catalyzes. And so the idea is that sequential application of these enzymatic reaction rules allows us to reconstruct the entire chemical structure of this genetically encoded small molecule. So how well does this approach work? Well, we codified it in a knowledge base web application called Prism, which is still around today. And we were able to show that Prism actually works pretty well. It's still, I believe, the most accurate platform to predict the chemical structures of genetically encoded natural products. It's also the most comprehensive platform. And in fact, we showed that Prism's chemical structure predictions were so accurate that we could even predict the likely biological activities of these genetically encoded small molecules from their predicted chemical structures with some significant degree of accuracy. And that was significant because it allowed us to systematically mine thousands of sequenced bacterial genomes for predicted antibiotic, antiviral, antifungal, immunomodulatory, and anti-cancer compounds. So now we have this vast resource of predicted structures and their predicted bioactivities. We now want to actually isolate and discover these predicted molecules and then go and test their activities. And so to do this, we developed a metabolomic analysis arm of this platform that we call GNP, which is designed to search for these predicted chemical structures in metabolomic data from the bacterium of interest. Like the structure prediction module, this is done in a rule-based manner. And here the rules are heuristics that we borrowed from the field of proteomics of enumerating all possible fragments of the chemical structure after breaking one or more labile chemical bonds, for instance, amide bonds, like I've shown here. So stimulating the breakage of all of these bonds allows us to enumerate the masses of a number of fragments that we might observe in a mass spectrometer if this molecule were indeed present. So you can think of this as being like a fingerprint or a barcode for this predicted chemical structure, which we can then match against the experimental data. And the idea is that these fragments are actually sufficiently unique that we only really need to see a few of them in order to identify this predicted chemical structure. And that is very useful because it means that our chemical structure predictions don't need to be perfect in order for us to actually isolate this compound that we predicted based on genome sequence information. So this combination of mining bacterial genomes for predicted chemical structures and then finding those predicted chemical structures with metabolomics ended up being quite powerful. It allowed us to target new small molecules for discovery based on genome sequence information and then automatically isolate them for further characterization. And so this technology platform helped us discover dozens of new bioactive metabolites, including some produced by extremely well-studied bacterium that had evaded detection because they were so low in abundance, like this anti-cancer small molecule that I'm showing here. So that was very exciting, of course, and this was a really rewarding line of work, but it also has a number of important limitations that I wanted to touch on in the context of what I'd like to now switch gears to talk about. And I think probably the most important and obvious of these limitations is that for any given set of chemical reaction rules, there will always be chemical structures that we cannot generate using those rules. So a very obvious case of this is in the instance of a novel enzymatic reaction that is discovered to catalyze new chemistry for the first time. And this was actually the case in the example that I'm showing at the right. This is a small molecule called streptide that has this very unusual lysine tryptophan linkage. And when this compound and the corresponding enzyme were discovered and characterized, we added this enzymatic reaction rule into PRISM. But of course, we could have never predicted the existence of this structure prior to that. And then a very similar limitation applies to predicting the mass spectrometry data from chemical structures. So in other words, for any given set of bond cleavage rules, there will always be fragments that we cannot explain by breaking those labile chemical bonds. And you might think that we could get around this limitation by being a little bit less selective in what chemical bonds we consider for cleavage in the first place. But we very quickly run into a problem of combinatorial explosion where we're generating so many fragments of this parent structure that we could match them to essentially any mass spectrum in our metabolomic data set. So after this work with Nathan, I wanted to expand my scientific horizons. And so during my MD and my PhD, I actually ended up moving away from small molecules altogether into proteomics. I spent several years with Leonard Foster at UBC mapping protein-protein interactions using an assay called cofractionation mass spectrometry. And I also spent some time working on single cell and spatial genomics with Grégoire Courtine at EPFL, where we studied the biology of spinal cord injury. But the whole time I was working on these problems, I really couldn't shake this growing feeling that actually small molecule mass spectrometry was the most exciting thing in computational biology. And so also during my PhD, I started to become interested in how these emerging technologies from artificial intelligence could perhaps overcome some of these limitations of rule-based approaches that I just outlined. And so the work that I'm going to tell you about now was done in collaboration with David Wishart at the University of Alberta, who's been a leader in this field of metabolomics for many years. So David and I had this thought of taking these AI tools that had been developed for human language and applying them to textual representations of chemical structures. And these are typically represented using a format called SMILES, an example of which I've shown below, which has actually been around since the 1980s. Now, obviously, these language models have attracted a huge amount of attention lately, but really for all this hype, I think the core concept behind how they work is just remarkably simple. Of course, the basic premise of a language model is to learn to predict what the next word in a sentence is going to be given the ones that have come before. And so, for instance, a good language model should learn that a sentence that starts with the words "the clouds are in the" is relatively likely to end with a word like sky or maybe air, and much less likely to end with a word like school or popcorn. And so we thought that instead of training one of these neural networks to predict the next word in a sentence, we could train the exact same kind of neural network to predict the next character in a smile string. And our idea was that after we trained this neural network, we could use it to sample new smile strings and therefore new chemical structures of plausible metabolites from this language model. So to break this down a little bit more concretely, we train this chemical language model by providing it with a very large training data set of chemical structures that we represent using the SMILES format. And for each of these SMILE strings, we go through the string character by character. And at each step, we ask the language model to predict what the next character is most likely to be, given the ones that have come before. And so at each step, the language model is being asked to learn a probability distribution over the tokens that comprise this SMILES language, again, based on the sequence of tokens that it's already seen. And then once we've trained the model, we can use a very similar autoregressive approach to sample new smiles and therefore new structures from this trained model. We do this by starting off sampling with this special start token and asking the language model to predict the next character. And we do this repeatedly, allowing it to build up this smile string step by step until it reaches the end token, which indicates that sampling is done. And the thinking goes that, our thought was that by doing this, we could use this language model to generate totally new chemical structures that we could then target for discovery in human tissues or biofluids. So again, the idea is that these models allow us to take a training set of molecules, train a neural network on their chemical structures, and then use that trained neural network to sample entirely new small molecules from the same areas of chemical space as the training set. And importantly, unlike this previous line of work on bacterial metabolism, this would be done solely by learning the statistical properties of the SMILES representation without having to define any rules for what makes a valid chemical structure, how we might generate valid chemical structures, or how metabolite-like these chemical structures are. And we thought that we could then search for these generated structures in mass spectrometry-based metabolomics data. Very early on in this work, though, we realized that we would first have to address a major technical challenge. The vast majority of studies that had worked with these kinds of chemical language models almost exclusively in the setting of drug discovery had trained these language models on hundreds of thousands of small molecules. And this had worked really well, but in the setting of human metabolism, there just isn't a training set of millions of known metabolites. There are at most tens of thousands, depending on how exactly you define human metabolite. And when we first tried to train a chemical language model on just tens of thousands of SMILE strings, we found that our model could barely even learn the syntax of the SMILES representation. And so we first had to figure out whether it was even possible to learn a chemical language model on such a small data set of chemical structures. The solution that we found is a new twist on a very old idea called data augmentation. And so if you're not familiar with this term, the basic concept is to create many slightly different copies of one's input data. So in the setting of image processing, for instance, if you wanted to train a neural network on photos of cats, the idea is that you can crop and rotate and mirror those photos. At the end of the day, it's still the same cat. And there's a nice analogy to chemistry in that there is actually a number of different ways to represent the exact same chemical structure using the SMILES format. So by convention, every small molecule has a single unique canonical SMILES representation. But it's actually straightforward to generate dozens or even hundreds of non-canonical SMILES that all represent the exact same chemical structure. And we found that when we started feeding many of these non-canonical SMILES into the neural network, we could essentially trick it into thinking that it was learning from a much larger data set of chemical structures, to the point that all of a sudden, a neural network trained on just 1,000 small molecules was almost as good as one trained on 100,000. And this ended up being the key insight that allowed us to start actually using chemical language models to discover new small molecules with mass spectrometry. And I told you that we first set out to do this in the setting of human metabolism, but actually our first successful proof of concept came in a very different context in the setting of forensic chemistry, where we used chemical language models to discover emerging designer drugs of abuse. And this was work done in collaboration with Peter Dalsgaard from the University of Copenhagen. So what exactly are designer drugs? Typically, they're slightly modified versions of well-known illicit drugs like methamphetamine or fentanyl that have been slightly modified from a chemical perspective in order to create a derivative that has all of the psychoactive properties of the parent drug, but avoids drug control laws. And so you may be familiar with some examples of designer drugs, such as synthetic cannabinoids, synthetic fentanyl derivatives, or synthetic cathinones, which are better known in the media as bath salts. And because these drugs are specifically designed to avoid drug control legislation, there are literally dozens, sometimes hundreds of new drugs emerging on the market every year as new laws are introduced to ban the previous wave of designer drugs. But just like with natural products, actually identifying these small molecules is quite time-consuming and labor-intensive. You have to identify what could very well be a completely unknown small molecule in a complex mixture that contains thousands of distinct compounds, such as human blood or urine. And so we asked if chemical language models could help accelerate this process. We obtained a training set of about 1,700 known designer drugs, and we trained a chemical language model on these structures, which we represented in the SMILES format. And then we started sampling new chemical structures from this trained model. And we found that using this non-canonical SMILES trick, we were able to train a model that could sample new chemical structures from the exact same areas of chemical space as the known designer drugs in our training set. So these plots that I'm showing here are showing a two-dimensional representation of chemical space, and each point in these plots is a chemical structure. And you can see that there's a very strong correspondence between the generated molecules on the left and the known designer drugs on the right. And qualitatively, this suggested that our model had learned to generate molecules that were similar to known designer drugs. Of course, we also wanted to test that idea more quantitatively, and to that end, we asked whether our model could successfully generate the chemical structures of designer drugs that emerged on the illicit market in the 6 months after it was trained. So we trained our language model, waited for another few hundred designer drugs to emerge on the market, and then asked whether our language model could anticipate the emergence of these drugs. And remarkably, we found that our language model correctly generated more than 90% of these emerging designer drug structures. One thing that we noticed while we were doing this experiment, and perhaps the key observation in this paper, was that not all of these molecules were equally likely to be generated. So as we looked through the output from the language model, we started to notice the same molecules were being generated over and over. And so to quantify this more systematically, we drew a very large sample of SMILE strings from our trained language model. We generated 1 billion SMILE strings. And we then calculated how frequently each unique novel chemical structure appeared in this output. And we found that the vast majority of unique chemical structures were only being generated once or twice, again, in this sample of 1 billion SMILES. But on the other hand, there was a small minority of structures that were being generated hundreds of thousands of times. Shown at the top left and bottom right, respectively. So of course, we were very interested in these frequently generated novel structures, and we started to look at them, and we immediately noticed that the single most frequently generated novel molecule was this structure that I'm showing here. And it turned out that this structure was actually a new synthetic cannabinoid that had been discovered less than a month after we finalized our training data set. We also noticed that more systematically, it seemed that the more frequently a novel structure was generated, the greater degree of chemical similarity it tended to demonstrate to a known designer drug. So in other words, structures that were generated more frequently were more designer drug-like. And so these observations led us to wonder whether this frequency was some kind of metric that could be used to anticipate the emergence of designer drugs most likely to hit the illicit market next. And we tested this idea on a new designer drug that the Danish National Forensic Lab had just discovered. This is a new derivative of the well-known street drug PCP that they had named deoxymethoxetamine. Because this drug had just been discovered, there was no possibility whatsoever that there was any kind of leakage with our training data set. This was a totally novel compound. And we did a thought experiment where we asked if all we knew about this totally novel drug was its exact mass, what structures would our model have proposed for this unidentified drug? There were about 18,000 generated molecules in the sample of 1 billion SMILE strings that could have explained this exact mass, plus or minus some small window of error. But these were not all equally likely to be sampled. And remarkably, the single most frequently sampled chemical structure was that of deoxymethoxetamine itself. So we thought this was pretty remarkable. Again, this is a totally novel drug, and given really minimal analytical data about this drug, our model had spontaneously guessed its exact chemical structure. And not only that, but all of the other top-ranked candidates were either variations on the same chemical scaffold, that of PCP, or they were highly plausible predictions in their own right. For example, structure number 3 here is actually a synthetic cathinone that has previously been detected in human urine. Here I'm showing you the results of a very similar thought experiment that we did with a synthetic cannabinoid called ADB hexanaca. And at the time that we did this analysis, this was the most recent synthetic cannabinoid to have emerged on the illicit market in the United States. And what you can see here is that not only is the language model correctly prioritizing the exact chemical structure of ADB hexanaca from its exact mass alone, but also that all of the top-ranked chemical structures are really variations on this exact same scaffold. And so I think it's remarkable that the model is able to identify the exact combination of decorations to this cannabinoid scaffold that's most likely in the context of known synthetic cannabinoid chemical space. So it seemed to us that our model had learned some sort of statistical function over the structures of unseen designer drugs. And we started to refer to this as the structural prior. And we called it a prior because we thought that the evidence from the language model could then be combined with more detailed experimental data acquired by mass spectrometry, specifically tandem mass spectra or MSMS, and that this would really enable full structure elucidation of totally unknown designer drugs with metabolomic data. And indeed, we found that when we combined the language model structural prior with data from tandem mass spectrometry, we could perform structure elucidation of totally novel designer drugs with a remarkable degree of accuracy. So on a set of 79 designer drugs that were discovered after we finalized our model, we could get the exact chemical structure right for 51% of these. And to give you some context for how difficult this is, the same test set yielded an accuracy of just 1% with mass spectrometry alone. We also found that our language model ranked the correct chemical structure in the top 10 almost 90% of the time. And this was an exciting observation because it suggested that 9 times out of 10, the correct answer would be somewhere within our model's short list of just 10 compounds, which is a very reasonable, manageable number of structures for a toxicologist or a forensic scientist to evaluate. And we noticed that even when this language model failed to predict the correct chemical structure, it often was predicting something very close to the ground truth. So for instance, here I'm showing you a new designer analog of the drug MDMA or ecstasy, and the predicted structure on the right. You can see that the model is getting the core structure right, and it's really just misplacing a single methyl group. And so this is quite a reasonable prediction. So this work ended up getting quite a bit of attention in the media, which was very exciting in that it led to a series of collaborations with forensic labs and law enforcement agencies who are very interested in using this kind of chemical AI to actually discover new designer drugs in their samples. And one collaboration that was especially rewarding was when we were approached by a major US law enforcement agency in the middle of a mass casualty event, who asked us if we could help them identify the causative designer drug. So again, I think this is an example that really just underscores the real world implications of computational mass spectrometry for small molecules. And some of these technologies are now actually implemented in the routine clinical workflows in one Canadian tertiary care center, and they've been used to make a number of discoveries that were not possible using that lab's existing assays. For example, one particularly noteworthy discovery was that of a new synthetic opioid called fluorofentanyl that we found had actually been proliferating quite rapidly within the population, but this was being completely unnoticed by public health officials and acute care physicians because the lab was simply not testing for this drug. Okay, so I started off by telling you that David and I were interested in using these chemical language models to discover new human metabolites. And after this long detour into forensic science, we have subsequently gone on to show that we can successfully apply the same broad approaches to model the human metabolome and to discover new human metabolites. So we trained a chemical language model on the structures of known human metabolites, and then we started using this trained language model to sample new chemical structures from the same areas of chemical space populated by the known human metabolome. And again, when we looked at the frequency with which these novel chemical structures were being generated, we found that the vast majority were only being generated once or twice or a handful of times. But again, there was this long tail of novel structures being sampled thousands of times. And remarkably, we found that these frequently generated structures were disproportionately likely to be known metabolites from a held out set that we had deliberately not trained our model on. We also again showed that the more frequently a novel structure was sampled, the greater degree of structural similarity to a known metabolite it tended to show, that is, the more metabolite-like it was. And again, we found that we could use the language model to prioritize the structures of unknown human metabolites from mass spectrometry data. So here I'm going to show you what this looks like if we assume we've observed a neutral mass of 176.0950 Daltons, plus or minus an error window of 10 parts per million. And all of the structures that I'm about to show you were deliberately withheld from the training set in order to simulate the appearance of a novel human metabolite. There are about 2,000 generated structures in the output from the metabolome language model that could explain this exact mass. But again, they're not all equally likely to be sampled. And the single most frequently generated structure is that of 5-hydroxytryptamine, better known as serotonin, which of course is an extremely well-studied human metabolite and therapeutic target. And the second highest ranked structure is also that of a known human metabolite. This is cotinine, the major metabolite of nicotine, and insurance companies around the world test for the presence of this metabolite in human biofluids every day. And most of the other top ranked structures are variations on these same two scaffolds, some of which are also known human metabolites, but are less well studied than these first two. So again, the key point here is that given as little information as an accurate mass measurement, this model can produce a ranked list of hypotheses about the identity of an unidentified or even unknown metabolite. So we repeated this analysis systematically for every structure in our training set in cross-validation, and we found that the model was able to prioritize the exact chemical structure of a held-out metabolite with an accuracy of about 30% from its exact mass alone. And also that the model ranked the correct structure in its top 3 predictions roughly 50% of the time. And this was remarkable, and both of these numbers were substantially better than the performance of a much simpler baseline model. We also found that our model was returning well-calibrated confidence scores for each of these structure assignments. So in other words, if our model thinks that there's roughly a 60% chance that it's generated the correct chemical structure for a given exact mass, we found that that structure does indeed turn out to be correct roughly 60% of the time. So this was a pleasant surprise, and you can imagine this is a very useful property for the model to have. Up to this point, I've been speaking in terms of neutral masses. Of course, in mass spectrometry, it's not always unambiguously clear what neutral mass has been detected because of the presence of multiple positively or negatively charged adducts. But we found that we could simultaneously consider multiple positive or negative adducts for any given held-out metabolite, and this had a relatively minimal impact on the accuracy of the language model. which again was a pleasant surprise. So these are very encouraging results. And again, we wanted to ask if we could integrate the language model with data from tandem mass spectrometry in order to perform structural elucidation of unknown metabolites. And using a relatively crude approach to predict tandem mass spectra for each of these generated structures, we found that we could improve upon the performance of the language model alone to identify a held out metabolite with an accuracy of almost 50% and a top 3 accuracy of 66% in both ionization modes. So up to this point, I've told you about how this language model seems to be quite good at assigning chemical structures to unknown metabolites that are observed by mass spectrometry. We also found that we can do the exact same analysis at the level of molecular formula instead of chemical structure. And we felt that this could be very useful because there are a number of applications of mass spectrometry in which tandem mass spectra are not actually collected. And so the highest confidence level at which one can annotate small molecules is at the level of chemical formula. And these include applications like imaging MS, as well as direct infusion MS. So here I'm showing you what this looks like given a different exact mass as input. Now we're looking at a exact mass of 325.102 Daltons, plus or minus a 20 part per million error window. And it's important to emphasize that there are literally thousands of valid chemical formulas that could yield this exact mass. But this language model trained on the known human metabolome seems to think that there are really only 2 plausible biogenic formulas here. And indeed, both of these formulas are either those of well-studied human metabolites or very plausible derivatives of known human metabolites. For instance, N-glycolyl neuraminic acid on the left, or this acetylated derivative of inosine that I'm showing on the right. So that was an example in which the model feels that there are 2 distinct formulas that could account for that observed mass with roughly equal accuracy. But in general, the model is quite good at assigning a single formula to these accurate masses with quite unambiguous confidence. And in fact, it's able to do this with an accuracy of over 99%. And so again, this is much better than the performance of a simple baseline. And just like in the structure-based analysis, these formula assignments are well calibrated and they're robust to considering multiple positive or negative atoms. And so we've actually now used this model to discover several new human metabolites that were hiding in plain sight in published metabolomics data. So for example, we focused on a data set of the blood metabolome in patients with end-stage renal disease. And we identified this novel brominated derivative of the metabolite niacin, which we then confirmed by ordering the synthetic standard for this compound and showing an excellent match between the synthetic and experimental MS data. Okay, so far I've told you about how these chemical language models are allowing us to discover new designer drugs and new human metabolites. And these successes had us wondering whether this is a more general property of chemical language models and whether we could develop this sort of structural prior for any domain of analytical chemistry. Of course, metabolites and designer drugs are not the only 2 categories of small molecules that are routinely analyzed by mass spectrometry. And so more recently, we've been testing this by training chemical language models on data sets of food-derived compounds, environmental contaminants, and then coming all the way back full circle to natural products. And this work has established that it's possible to develop surprisingly accurate structural priors in all of these domains of analytical chemistry. So the plot at the right is showing you the accuracy of a chemical language model at assigning complete chemical structures of held out natural products from their accurate masses alone. And it's able to do this with an accuracy of about 5%. So that's obviously quite a bit lower than we saw with the designer drug and human metabolome models. But I still think that that's actually quite a remarkable level of performance, simply because natural products are much larger and much more structurally complex and more chemically diverse. And so there's simply an exponentially larger number of potential chemical structures to consider for any given exact mass. And it's also much better than searching by accurate mass in PubChem, which is still a reasonably prevalent approach to annotating metabolomic datasets. And the performance tends to be even better for these food-derived compounds and the environmental contaminants. And that makes sense because these tend to be less complex and less chemically diverse. Again, even when the language model is wrong, it does tend to prioritize a chemical structure that's very close to the truth. So this plot is showing you that the natural product language model prioritizes structures with an average Tanimoto coefficient of 0.23 to the true held out chemical structure. And that may not sound so overwhelming, but in fact, that's a higher degree of similarity than more than 99% of pairs of small molecules in our training set. And it's also much better than searching in PubChem, which tends to yield structures that really aren't that much more similar than random expectation. So to sort of show you an example of this natural product model in action and kind of maybe illustrate why this is a more difficult problem than the human metabolome, here I'm showing you a very similar experiment to the ones that I showed for designer drugs and human metabolites, but here showing the indolocarbazole antibiotic K252A, which has been held out of our natural product training set. And when we search the language model output for molecules that match the exact mass of K252A plus or minus some margin of error, we get about 300 candidates, and one of these is generated much more frequently than any other. And gratifyingly, this turns out to be K252A itself. And again, we also see that all of the other top-ranked candidates tend to be variants on the same indolocarbazole scaffold, and we have to go quite a ways down the list before we start getting into more exotic structural territory. I told you that even when this model is wrong, it tends to prioritize a structure that's very similar to the ground truth. And so an example of that is the case of numecandin B0, which is the precursor to a clinically approved antifungal drug called caspofungin. And so if we search in the language model output for the exact mass of this chemical structure, we see that there are about 3,000 candidates, and they're sampled somewhat more uniformly than we saw for K252A. But nonetheless, we see that the top-ranked chemical structure is almost identical to that of numecandin B0 itself. The only difference is that the language model is proposing a linear acyl chain rather than the branched one in the true chemical structure. And again, the other top-ranked structures are either variations on the same scaffold, or else they're very plausible predictions in their own right. For example, structure number 3 is actually very similar to a known cyanobacterial microsystem. So to kind of wrap up what I've shared, I think that this line of work has shown that chemical language models are not only able to generate new chemical structures from the same areas of chemical space as the training data set, but also that these models learn some inherent concept of the plausibility of any given novel structure in the context of that training set. And that can in turn be used to prioritize totally unknown chemical structures from really minimal analytical data. And I think that that's a very exciting observation for 2 reasons. The first is that this is really a totally distinct source of information from anything that's currently being used to annotate metabolites or solve chemical structures. And so I think that chemical language models could potentially provide a springboard to really increase our accuracy on both of those tasks. And the second reason is that as I showed you with a number of these examples, these language models now allow us to think about a scalar value, the mass to charge ratio that we obtain by mass spectrometry, as a very rich distribution over plausible biogenic chemical formulas or chemical structures. And I think that that will be quite an important and useful tool in our ultimate efforts to extract more chemical information from mass spectrometry-based metabolomics data. So I want to thank, of course, a huge number of people who have contributed to all of this work over the years, in the McGarvey lab, the Foster lab, the Wishart lab, and the Dalsgaard lab, as well as our collaborators in the BC Provincial Toxicology Center who have helped us validate a number of these new designer drugs, and then a series of funding sources that have supported these different lines of work over the years. And of course, I'd be very happy to take any questions.
Thanks so much. So if someone have if anyone has any question, please raise your hand. And when you're chance comes, please unmute yourself and ask your question. Alternatively, you can post your question on the Zoom chat. So Dave, please unmute yourself and ask your question.
Sure, can you guys hear me okay?
Yep.
Okay, so my question is, I know one of the metabolites that you had shown was actually a monosaccharide that's commonly in glycan structures. Do you know anybody who's doing this language model-based approach on glycan sequences and glycan structures specifically?
Yes, that's a little out of my wheelhouse, but I believe Daniel Bohar, if I'm pronouncing his last name right, is working on that. I think he's taking a different approach to represent glycan structures than SMILES.
Oh yeah, they have like 24 different sequence formats. So that was another question I had. Do you know any language models that deal with multiple languages representing the same characters or words?
In the setting of chemistry specifically? That's an interesting question. Yeah, I can't say that anything is really coming to mind there, but that's an interesting question.
Okay, yeah, I figured it was kind of out of left field, so no worries.
So if no one has any question, oh yeah, I think I have a couple in the chat. So Mernush is wondering whether you have compared your results to online accurate mass search engines like Metlane or Medfrag.
Yeah, good question. I mean, I think that the sort of most direct answer is that those approaches will, by definition, yield an accuracy of 0% in the case that we're truly interested in, and which we're simply trying to approximate in cross-validation, which is the discovery or detection of a totally unknown metabolite. So those could be useful benchmarks simply in terms of asking, realizing that those approaches will definitionally yield an accuracy of 0%. How close can you get in terms of chemical similarity to the ground truth? But again, they're really those approaches are not able to suggest totally novel structures, which is, I think, the key gap that this language model is filling.
So Kelvin is wondering, does your model use predicted enzyme activities for uncharacterized enzymes from genomes? And the second question is, what percentage of activities are currently characterized, and what are the implications for complexity currently still being missed?
Another good question. Yeah. So the goal is really to sort of learn the biosynthetic logic that's just embedded in the structures of known metabolites, without directly modeling biosynthesis itself. As we have gotten more, confident in this model's performance and actually now used it to discover more than a dozen new metabolites, this is becoming a very relevant question. Where are these new metabolites coming from, and then also what are they doing? And so this is something that we're starting to think about, whether there's an opportunity to use protein sequences or structures, either within the same modeling framework or perhaps in some downstream complementary approach. But the sort of straightforward answer is currently no, it's not using predicted enzyme activities at all. It's really just trying to learn the kind of language of metabolite structures.
So if there's no question, please give Dr. Skinnider an online round of applause. And thank you everyone for being here. Have a good weekend.
All right, thank you for having me. Have a good weekend all.